today, who is Dr. Rebecca Schmidt-Jeffris, research entomologist with USDA ARS, in Wapato, Washington. Her research program focuses on the biological control of pest arthropods in tree fruit by their predators and parasitoids. She's the lead editor of American Entomologist magazine and a member of ESA Pacific Branch and Plant Insect Ecosystem Section executive boards, and was recently recognized as a Service to America medal finalist. Rebecca, welcome, and you're up.
All right, thank you so much, Steve, for the invite, and thank you everyone for joining us. If there's somebody that you know that really wanted to see this and couldn't make it this time for whatever reason, this will be recorded, as Steve mentioned, so it will be available afterwards. I also know some folks don't like asking questions, especially when you have to mute and unmute on Zoom. So if someone would prefer to send me a question via email, it's right there, and I'll also have that available at the end of the presentation too, if that's how you prefer to do Q&A. So what I'm going to be talking about today is a series of studies that really took off due to funding both from the Western IPM Center, which is why I'm speaking to this group today, but also from the Washington Tree Fruit Research Commission. So it's kind of starting out as a big thanks to both of those groups for supporting this research. So I'm going to focus mostly on the apple portion of what we've been doing and on two natural enemies in particular, but we have been working with additional natural enemies beyond the ones that I'll be showing you during this presentation. So if there are any questions you have about other natural enemy groups that you're not seeing, please speak up and I'd be happy to share some of that information with you as well. So I wanted to start by providing some context for those who are not tree fruit growers in the state of Washington or elsewhere in the Pacific Northwest. So while apples are considered a minor crop throughout the United States, that almost is not true for Washington. So we grow easily the most apples of any state in the United States by a wide margin, with nearly 60% of the apples in the US being produced by acreage within Washington. And that really becomes very striking when you look at this map, which is pulled from the Cropscape database. Usually you can't see a minor crop zoomed out at this level, but the apples are shown in that dark purple mauve color. So it's just a tremendous amount of apple acreage going all the way to the southern border, all the way up north to Canada, kind of following the main waterways for irrigation purposes. Our industry, in addition to being very, very large for a specialty crop industry, is special in a few other ways. So first, the climate is very different from other parts of the United States and indeed parts of the world where apples are grown. So this is a picture of sagebrush steppe, and this is kind of what we have out by the rivers in areas that aren't irrigated. It's very, very dry. What this means is that we do not have to manage plant pathogens nearly to the extent that in other areas where apples are grown, both in places like Europe, like the UK, but also in the Eastern United States. And so that kind of leaves us free to both focus more on insect control and therefore biological control, but it also means the apples are not getting sprayed nearly as much, which again opens the door for conservation of biological control agents in orchards. The other big way in which Washington apple differs from other places in the world is because of that large contiguous acreage that I showed in that map. Mating disruption for control of the number one pest, the codling moth, became adopted at a wide scale with, I think we're at about 95 to 99% adoption, depending upon which set of numbers you use. And again, this has allowed for a decrease in insecticide use for control of that pest, again, opening the door for focusing more on biological control because fewer pesticides are being used. That also has opened the door for lots of organic acreage to be planted. So about 20% of Washington's apple acreage is organic, making up more than 90% of the worldwide organic apple industry. And again, using organic chemistries, using them less frequently, and using fewer broad spectrum pesticides has really increased the focus on biological control. And then finally, this industry has a demonstrated efficacy for biological control. There have been a series of researchers whose shoulders we stand on when we do this research that have demonstrated various natural enemies play a major role in controlling pests, and therefore our growers understand and know that they can rely on these natural enemies and are more likely to reach for those tools than maybe other grower communities. However, stuff still happens out in the orchard, and there are times when our growers don't have enough predators and start to see problems with specific pests. So for a very long time, the greenhouse industry has been able to purchase natural enemies for release and then go ahead and release them in their greenhouses. And then because this industry existed primarily starting in Europe and then slowly moving into the United States, this allowed our apple growers to also have access to these products. Here's just an example of one where you can purchase a lacewing product online. However, greenhouses, which again is the main focus of the industry, where they're growing natural enemies in mass and then having them available for purchase, that's their primary audience. They buy the most product, and therefore those are the natural enemies they rear. And so I usually say I've got a lot of greenhouse envy. So augmentation, purchasing and releasing natural enemies tends to be a lot easier in greenhouses. There's one really obvious reason, is that it's an enclosed environment. So things that you release in a greenhouse are more apt to stay in the greenhouse than releasing them in an open air crop like tree fruit. The crops in greenhouses tend to be smaller. So the per acre rates in terms of scaling to a plant level, you can more easily say saturate the plant with your natural enemy as opposed to releasing it in a tree. Greenhouses are climate controlled, and they have a tendency to be more on the humid side, which is the opposite of our very hot, dry apple growing region in Washington. And this becomes a problem for us because the available species are typically reared with the pests of the greenhouse industry and the environment of the greenhouse industry in mind, which means a lot of them do not have the heat tolerance that some of our native natural enemies do, at least not to very, very high temperatures that we can hit. It also means that a lot of them are not arid climate adapted. And then because of all of that, and because of who the main purchasers are, when insectaries are developing recommendations for how their products should be used in things like per acre rates, they are making those choices and doing most of their R&D with, very reasonably so, the greenhouse industry in mind. So that it means that the recommendations that we are primarily getting from the insectary industry do not translate well to orchards. So that's kind of where we stepped in. And so one of the things we did with our Western IPM Center funding was conduct a needs assessment of our growers, primarily focusing on apple and pear growers in Oregon and Washington. So we had 132 growers respond to this survey, representing about 44,000 acres. A third of those growers had indicated that they tried releases on their orchards at least once, and that of those that they had treated on a fairly regular basis, it was about 8,000 acres per year. In terms of what was being purchased, the most common responses were lacewings, lady beetles, and predatory mites. The growers that were doing regular releases indicated they spent about $150 per acre on average. And then for us, the most important question was, if you are doing releases and don't think you're as successful as you could be, or if you're not doing them, what is your biggest barrier to success? And we had over 50% of our respondents say their biggest barrier was the lack of knowledge on best practices to doing releases. And fewer than 15% gave the answer of cost or perception that it didn't work combined. So this is indicating to us that we have an opportunity here, because I can provide increased knowledge in terms of doing research and giving them the information they need. I can't change the cost of the beneficials, and if they don't work, they don't work. So that's kind of where we've been directing our research program, is making these best practice recommendations based on research. The other thing that's important to note, and we'll see if we can get the video to play. It's pulled from online, so that's always a hit or miss. Yeah, we're doing it. Is that we've got a lot of drone use increasing in orchards for deploying beneficials. And this is a big opportunity for our industry in particular because of, as you can see from this video, the huge amount of acreage that needs to be treated. It's not like a greenhouse where you can go and disperse predatory mites with a little handgun and have one person go do that. If you're needing to treat a big 50-acre organic block of apples, that's going to be very labor intensive. So in order to save big on labor costs, people are starting to lean towards using drones, particularly those that are distributed by Parabug. So the growers have some really key questions on how to do releases. And it turns out that their questions are exactly the same as they would ask if a brand new pesticide came out on the market and the information they would expect to get from the label. What product should I be choosing? Which one works the best? When should I be applying it, both in terms of the time of year for efficacy and then how that lines up with the pest phenology? How many natural enemies should I apply, which from a pesticide label perspective, that would be the per acre rates. And then finally, how do I apply this, which is maybe a little bit more unique to our industry specifically in terms of drones or some of the other methods I'll show you. And then finally, the question that's not the same as a pesticide is how do we get them to stay? And this is a really tricky shift in perspective, even as a researcher, when you're thinking about how to space your plots apart, because you have treatments that can physically move into another plot if they so choose, because most of the things that we release can eventually fly. So I'll talk about a series of studies that is targeting two different pests. The first series of studies, the target pest was mealybugs. In Washington apple orchards, our main mealybug pest is grape mealybug, although occasionally there are some apple mealybugs in the mix. The big risk with mealybugs in apples is primarily due to honeydew damage to the fruit, causing sooty mold and streaking. There's also some concern about contamination of crops that are getting shipped potentially internationally, since mealybugs, as you can see from this bottom photo, like to hang out deep in that calyx into the apple, so there might be a risk of shipping that. Adequate pesticide coverage, even in conventional orchards, is hard for mealybugs because they are cracks and crevices species. They do this behavior of getting really deep into places that are hard for any pesticides to get to. And then in general, on top of that, our organic products are just not very effective for mealybug control. This is kind of a prime opportunity then for using a predator, which can get to, via walking and flying, the pest. So the thing we tried for these is mealybug destroyer, which is a ladybug species, a little bit smaller than what we're used to seeing, that is available, again, primarily for the greenhouse market. They can go from egg to adult in about 30 days. All the life stages, including the larvae, feed on mealybugs, and they're quite specialized to feed on mealybugs. The downside for us is this is a tropical, subtropical species, so it can't persist below 20 Celsius. Which means that if a grower wanted to use this, they would have to release them every single time they had a mealybug problem. They're not going to establish and then keep controlling the problem. A benefit of this species is that it is so specialized on mealybug, is it will actually sniff out mealybug wax secretions and honeydew, and those odors actually stimulate the adults to oviposit. With many of them actually ovipositing directly on top of mealybug ovisacs and kind of providing their babies with their first snack right when they're hatching. So the first trial that we did in 2020 was in really small plots, about 1/10 of an acre in a commercial orchard. And we wanted to test a few things. The first thing we wanted to test is what we would call a high rate of 5,000 adults released per acre. That was more of a kind of spot treatment recommendation from the insectary, not one they said that most growers would want to do through the whole block. A lower rate, which would be more of a whole block rate of 2,000 per acre. And then we looked at two timings. The first timing, that 14th of May date, was basically as early as we felt comfortable not risking a freeze and accidentally killing the adults as they were released in the orchard. The later timing corresponded to when this particular orchard started to see ovisacs. With the reasoning being that we thought that that would be a good time to release adults to get them to start laying eggs fairly quickly. And then we also, during that second timing, threw in a drone-based treatment using the high rate, also with a no release control. So first, looking at recapture of mealybug destroyer adults. So we used beet sheets. So we're just putting a beet tray underneath the tree branches in the orchard, hitting it, and then counting insects that fall on it. So here you can see what the adults look like as they drop onto that beet sheet. So what we found is right after that early season release, which is that first red arrow, we found quite a few adults in the treatment where we released them the first time and the second time we sampled. So we're seeing a lot of them in those first 2 weeks, and then really not seeing them too much again until after we did that second release late. And then recapture rates were not nearly as good with the late release as they were with the early release, likely because they weren't moving as much after that early release because of how it was much cooler at that point in May than it was getting into mid-June. The other thing I want to point out is, again, you have the low recovery rate of recovering about half of a mealybug destroyer per acre in that second set of releases. We also had rapid movement, especially in that second one, of the mealybug destroyers into the control plots, which is again a risk when you have insects that are highly dispersive and can fly right as you're releasing them. So those controls are shown in the white. We also never found any again inside the drone treatment plots. And I'll talk a little bit about that from this particular trial in just a second. In terms of pest control, so these are graphs of mealybugs recovered from these burlap strip traps, which are just burlap tied to tree limbs, and they crawl in there because they like cracks and crevices. The first graph is counting the moving mealybugs, the adults and later older juveniles. And the second one is counting the ovisacs laid inside these burlap strips. So there were no significant differences between any of our treatments statistically, but we did see kind of a trend in that right after that early release of having fewer mealybugs in that release treatment compared to the control. But then things got a lot more muddied after that later season release. But again, maybe some indication that all the ground or hand release treatments were kind of clustering together. So maybe a very mild effect, if anything. And this is reflected if we look at the seasonal sum counts, both from shoot samples and from those burlap strip traps, where we in general see lower numbers in all of the hand release treatments, but not in the drone and the control. So did the drone not work? We'll get in a little bit into what we were kind of figuring out with the drone at that point, but that was kind of our initial question. We also had questions from growers about whether or not our climate was suitable for the mealybug destroyers to even reproduce in the orchards. And the answer is yes, they can reproduce in orchards in which they're released. So in this trial, we only found a few mealybug destroyer larvae. When we were finding them somewhere between 40 and 70 days post-release, depending upon which plots and where we were finding them. So we could tell growers that if they do have a successful release, they can expect to see those larvae in about 6 to 8 weeks. So what are our take homes? Well, the first one you may have figured out is that I figured out we needed bigger plots. The 10th of an acre plot was just not big enough to do a trial with such a mobile insect. I've hinted a couple times that we thought maybe the drone was not working. However, right at this point is when the industry started realizing that there were people who were doing these releases of mealybug destroyers by drone, and they went, oh, so we're concerned about that because this insect exhibits a behavior called phototaxis. It is attracted to light. So if you're releasing them up high over a tall crop, they might just immediately exit the drone hopper and fly towards the sun, and you might be providing your neighbor several orchards over with biocontrol, but probably not the area directly below the tree. So okay, we'll change that up next time we do this. We have some indication that the early season releases might work better, since those were the ones where we did see more indication of differences. And we also found that lower release rate appeared to have the same general efficacy as the high release rate. So in 2021, we took all of that information in and we redesigned our trials. And we went to a spot that had previously lots of mealybugs in it. And they elected, as sometimes pests do in these trials, to not show up. So throughout the season, we tended to find about less than 1/10 of a mealybug per one of our strips. And because this predator is a specialist, that meant it didn't want to stick around because there was nothing for it to eat. So a day after releasing, we found less than 1 mealybug destroyer per acre. We did find 3 per acre in the ground treatment. And then 1 week after release, I think this represents 1 or 2 total mealybug destroyers found throughout all the trials. So again, maybe a little bit of an indication that those mealybug destroyers are not making it into the plots. For this trial, this is particularly an important indicator because in this one, we made sure to do the drone-based releases as it was dark outside. So that's not a phototaxis issue from the drone at this point, but it's a little hard to make this comparison just because there was, again, no food for them to eat. So again, we took that all in and we went to another orchard that had higher mealybug destroyer population or higher mealybug populations. And we also at that point managed to find a source of the larvae. to see if maybe releasing a stage that can't fly would help it provide biological control before they molt into adults and then fly away. So we did the adults and larvae as ground releases. We released the adults by drone. And right as we were finishing up our trial, the grower came in with a fire blight spray. Fire blight is a bacterial disease, so it's not an insecticide that was sprayed, and it is an organic orchard. But there have been some indications from other growers that with concern about toxicity of sprays that go on for fire blight in organic orchards and whether or not those need to be more precisely timed to avoid non-target effects. So that's a possible problem that happened in this orchard. It also was a big indication to us because of how important doing these early season releases is for good biocontrol, that we really need to start working on developing a program that allows growers to integrate these releases with early season disease management, because you can't just not manage for fire blight, it kills trees. So that's one of our upcoming research questions that we're hoping to work on. So because we're gluttons for punishment, we did this one more time. So in 2023, we picked an orchard that is really well known for having lots of mealybugs in it. And we also had a grower who had been releasing natural enemies in several of the other orchards they work in before and had kind of a standard program that they like to use. So in this one, we compared our mealybug destroyer releases with what we call a grower standard of releasing convergent lady beetles, and then also a no release control. And once again, we did not see any differences between what we released, what the grower released, and the control in terms of mealybug counts, either the adults or the ova sacs. Now, I will say we did have a pretty quick dip in mealybug numbers. So they started out looking pretty good and doing good for doing a pest control related trial, and then kind of dropped rapidly late in the season and sort of disappeared kind of on their own in this orchard. But again, they were around long enough post-release that I think we feel pretty comfortable saying that neither of these natural enemies did anything. We also found no destroyers or convergent ladybugs post-release, with kind of one caveat is we found one dead mealybug destroyer after we released them. And we found 3 of convergent lady beetles. But that orchard also had convergent lady beetles in it went before our release. They're established and native in Washington. So there's no way to know whether or not they were the released ones or the ones that were just already native. But this is kind of an indication that neither one of them did anything and they probably didn't stick around. So my general consensus with doing biocontrol augmentatively in tree fruit is that it just does not work well with adult ladybugs. They are so dispersive that it's going to be very difficult to figure out ways to get them to stay in the orchard and to assess whether or not they're doing successful pest control in the spot where you released them. In terms of mealybug destroyers, because we saw only the 1 year where there was maybe an inkling where they had an effect, and because this is the price of releasing them per acre, depending on who you buy them for and what rate you use, I think my recommendation here is don't do it and save yourself this cost per acre, and let's try to figure out something that actually works, that's not nearly so expensive. So I didn't want this talk to be all doom and gloom. So we're switching to a natural enemy that has a lot more promise for working well in Washington, and that's releases of lacewings. So right now there are 2 species available on the North American market that people can purchase. There's Chrysoprilla rufilibris. This is by far and away the more common one folks purchase. My current understanding is we really don't have any growers purchasing the other species. And that's also the most common one that people are using in greenhouse industry and really all other industries in the United States. This is kind of the big market one. Chrysoprilla rufilibris is not native to Washington. So this is its existing range map. I'm sure these 2 are just not colored in because no one's looked. But it's pretty clear that it's not coming west of the Rocky Mountains. And it's never been found despite lots of work surveying lacewing species in Washington orchards. So it also looks like it can't establish either. The other species listed on the market is Chrysoprilla carnea. This species is potentially native, and I'll get into why I'm saying potentially here in a minute. It might be a better climate match. There's some research indicating that it's a little bit more tolerant of arid and hot places, but it is a bit more expensive than Chrysoprilla rufilibris, which is why people tend to purchase that one as opposed to this one. In terms of how you can purchase lacewings, all 3 life stages are commercially available, especially when you're looking at, again, the bigger market Rufilaris. So you can buy adults. Most people do not because they are much more expensive than the other stages. Adult Chrysoperla are not predaceous, which means that if you release them, you're waiting for them to lay eggs and you're waiting for those eggs to hatch before you see any kind of biological control happen. And then a lot of researchers, especially in the 80s and 90s, were discouraging folks from doing releases of adults because Chrysoperla also have an obligatory dispersal phase right after they molt and become adults. So they tend to disperse, mate, and then start laying eggs. And obviously you don't want your freshly purchased and released expensive biocontrol agent doing that. So no one is really using adults, and for good reason. The larvae are more commonly used. They are about 6 times more expensive than the eggs, but that is offset by having a generally lower release rate recommended for them than eggs. And that's because the thought is they're already hatched. They can kind of defend themselves. They can move around. You're not worried about maybe a poor egg hatch in a batch of eggs that you've ordered, so that you can drop that release rate. So the cost gets close to equivalent because of that lower release rate. Eggs are, as you can guess, the cheapest stage, but that does mean you are releasing eggs and then you have to wait for the eggs to hatch for biological control to occur. Now, some people get around that by holding the egg container at room temperature, slightly warmer, and waiting to see that they're hatching before they do their release. The other big risk with eggs is that if you're sprinkling a bunch of eggs around an orchard that has a lot of other insects living in it, that there is a risk of those eggs being eaten by those insects, or again, having non-ideal conditions for egg hatch to occur, and then decreasing the overall number of biocontrol organisms you're getting from your purchase. Lacewings can come in a few different methods for release. One of the ones that has been tried by a few orchardists is egg cards. So egg cards come as kind of one big chunk of cardboard. You can see here some segments. So you tear along those segments and hang them in the tree or in whatever plant you're using. There are some pretty clear pros for this. In terms of someone managing an orchard, it's really easy to give people instructions for using these. You know what your rate is. You say put 1 card every X number of trees as you go down a row to hit that rate that you want. They won't fall out of the tree. If you're sprinkling a loose carrier material with eggs in it into the tree, the odds of actually keeping all of that on the leaves or on the bark is really low. And these also allow you to target hot spots, which is kind of what we did here. You can see a leaf that's a little curled up because there is a nice aphid colony sitting right there. The cons are illustrated by these stop motion cameras that we were using to monitor these egg cards in the field. The biggest one being ants. You absolutely should not release really lacewings in general, but especially use these egg cards in an orchard that has high ant populations, which this one did. All of these little black smudges are ants. They showed up in less than 30 minutes and had this card wiped clean in under 24 hours. So they just do not work in those circumstances. There's also a risk that other predators may see these egg cards as basically like a little buffet and come and pull them all off before you have a chance to have them hatch. Here's an earwig that we think was doing just that. But we haven't seen or done a study yet to determine how common this is or how large the risk would be in a commercial orchard. There's also some questions about whether or not the first lacewing or 2 to hatch would then eat all of their brother and sister eggs on the card. But again, this is a research question that we haven't looked at yet. And then finally, the eggs are a little bit more expensive as a card than they are loose. So given all that information about what's on the market, what we do and don't know about what's on the market, we wanted to release these for our big target pest aphids. In particular, woolly apple aphid is a major pest of organic apples and can be quite difficult to control in conventional orchards, but is very difficult to control in organics. A typical organic orchard might see, if they've got a woolly apple aphid population, up to 5% yield loss. Depending upon the variety, that's $500 to $4,000 per acre. So this is a big issue. In addition to woolly apple aphid, we also have rosy apple aphid and green apple aphid as pests, and sometimes you'll see all 3 of them together in the same orchard. So we started doing these release trials with lacewings in 2021. We used 1/4 acre plots, and all of these are in commercial organic orchards. This first year, what we wanted to test were hand releases only, and we wanted to compare eggs of the 2 species that were available versus larvae. And you can see we did that 20% reduction in the rate recommended for the larvae. This is a little bit higher than what a grower would do. And a lot of our growers will do repeated releases of smaller amounts. We wanted to just test what one looked like to see if there was anything that showed potential for success. So this is kind of a representation of what hundreds and hundreds of thousands of lacewings look like when you need to do one of these trials and you get them all in the mail. So we did these releases really early in the season on May 5th, kind of again trying to avoid risk of frost. And you'll see I've got some of these here in quotation marks. And I've kind of mentioned that this might be something that we talked about here in a second. So the Rufilabrus came from one insectary. And they were as advertised. So we looked at the morphology and said, yes, those are indeed C. rufilabrus. Then we got to the C. carnea. And so the C. carnea were obtained by a distributor from two different insectaries, one to get the eggs and one to get the larvae. The eggs were as advertised. They come from C. carnea and sensu latu. So these cannot be morphologically identified to a specific species. You need to do some other special things to get the actual species ID, which I'll talk about here in a second. But the second one, the carnea larvae, we identified morphologically as C. externa. It is not in the C. carnea species group and was the incorrect species for what we were looking for. This mattered, and we'll see I eliminate this treatment from all of our future trials because C. externa has a southeastern distribution and is likely a very poor climate match for Washington, which is kind of what we were going for by testing this additional species. So here's kind of how folks identify lacewing larvae using morphology. It's all based on the markings on the head capsule and can get a little bit challenging, especially for a non-expert, in distinguishing some of these species, which look very, very similar apart. Fortunately, C. externa is one of the easier ones. So I mentioned C. carnea is a species group. It is not a true species unless we are talking about the one that is collected from Europe. So a lot of studies will label lacewings as C. carnea if they're within the C. carnea species group. But unless it was collected from the UK, it is not actually C. carnea. Instead, what we have in the United States that is in the C. carnea species group consists of these species listed right here. And in Washington orchards, it is these last three right here, Johnsoni, Pluribunda, and Adamsi. Lacewings are in this particular group are distinguished by species once you can't get further with the morphology by listening to two reproductive adults singing to each other. This is obviously challenging to do with a dead specimen, i.e. impossible. And it also means you have to have a male and a female of the same species and then be able to do the song analysis, which is actually the least challenging part of all of this. Most of the time, it's just getting the critter necessary to do this. So this is why this has been such a challenge for the lacewing industry in general to really be certain of what they're selling. So the next question folks always have, well, why don't you just do genomics work with them? And that is something that's been worked on by that same group that's doing the song analysis. So in general, they use these four gene regions, sometimes just a few in combination, but you usually need more than one. And so they've got this nice phylogenetic tree worked out based on the genetics and then also incorporating the song structure. However, if you've got a species that already hasn't been positively identified, you can't just look at where it is in this position on the tree because it looks like the pre-existing identifications of lacewings are polyphyletic, which means some of the genetics are not necessarily reliable. So again, it gets really complicated. So based on both our morphological work and then doing some CO1 sequencing and then what we know about what's currently actually being sold on the market is it looks like what we purchased, the C. carnea sensu latu, is actually C. pluribunda. So I will refer to it as that throughout the rest of this talk. So we've actually got C. pluribunda eggs and C. external larvae in this trial and comparing them to C. rufilabrus eggs and C. rufilabrus larvae. So what we found in that first year was very promising. So after one release, this is our release day shown here, we were getting statistical differences between treatments, keeping that control very consistently throughout the season, had higher numbers than most of the other treatments. And what we found was it was the C. pluribunda eggs and the C. ruffilibris larvae that seem to be the best performing and the ones that statistically separated from the check in our seasonal sums. And we're looking at a pretty decent reduction in aphid colonies with about 50% between the two treatments that seem to have an effect and just after just one release. So if we'd done it more, we might have seen an even stronger level of control. I want to point out that on our release day, the aphid counts were quite low. They were just starting to get going for the season at 0.04 colonies per shoot. So very, very few aphids. In terms of recovery of our lacewings that we released. So for all of these tables that you'll be seeing for these trials, that red highlight is the first date of capture for anything that we're talking about. I'll have the treatments at the top, and then a set for the resident Chrysoperla that were just native to the orchard, and the set of resident Chrysopa, which is our other genus of green lacewing that we get in Washington orchards. In this trial, we did not recover any adults of the species that we released. So we'll start by highlighting two that had really great recovery in terms of the number we recovered per plot, and both of them were larvae treatments, indicating that maybe we get better recovery and maybe we'll have greater success with larvae-based treatments. Then looking at the two treatments that were the most successful, the one thing that stands out to me is that we're still recovering them in low numbers, albeit 4 weeks post-release, whereas the others, we didn't get them past 3 weeks post-release. So maybe we'll find them later, and that's an indication of a more effective treatment. Then the one benefit from doing these releases on top of just kind of traditional control reasons and adding to the natural enemy population is that we appear to have gotten our lacewings out into the orchard 1 to 2 weeks before lacewings that were naturally present in the orchard had larvae that had, maybe the adults had come out from overwintering, laid eggs, and had larvae available to start doing biocontrol. So maybe we got a head start on the resident natural biocontrol in that orchard, and that's why we saw such a good effect. And then finally, one of the other reasons this trial likely worked so well is that our released populations were actually a substantial increase over that resident lacewing population. And in some cases, a 10% or over 100 times increase than what was naturally present in those plots. So we're really giving biocontrol a big bump in this particular orchard. So how do I know that what we recovered was what we released? Because I did say that it was complicated. So this is what the approach we've taken for these studies, which seems to be working well so far. We did a first set of this with just the 2021 data, and we just sent out our last set of sequences from the last 2 years that we've done to kind of finish up this work. So I mentioned that they built this phylogenetic tree using these 4 gene regions. And then I wanted to point out using that our Washington orchard lacewings tend to lie somewhere in this group, which is called the C. floribunda group. C. externa is somewhere down here. So that's that larvae that we ordered without meaning to. And then here's C. ruffilibris. So we've got some good separation between what we're looking for genetically. So what we did is just run CO1 to see if we could get away with only having to do 1 set of PCRs and still kind of figure out who was who for the larvae that we were recollecting. So we used one of our native green lacewings in the Chrysopa genus as our outgroup for this and constructed a phylogenetic tree for every single lacewing larvae that we recovered from this orchard. So what we did first is we grabbed some lacewings straight from the bottle. And in all cases, some of these are going to say eggs and some of these are going to say larvae. But what that means is it's the type of order it came from. We did wait until the eggs hatched. So we actually are testing larvae in all these circumstances. So we had from the C. carnea egg treatment, we had some straight from the bottle. And from each of these treatments, we had 4 to 5 individuals that came straight out of those bottles. And those are matching the color code that I have in this gray box. Then I'm going to add in the ones that we collected on specific dates from specific blocks that were linked to specific treatments and use that same color coding scheme for what treatment should have been in that plot. what you'll see is they all cluster super nicely with the treatments that they came from. So this is an indicator that this method we're using is actually quite helpful, and that I don't have to make my technician run 4 different genes and do 4 different PCRs for every single plate of lacewings we collect, which is much appreciated. So what we found is that we have this set that all matches from the treatment. So that's our recovery there, 2 individuals. And then here, each one of those is recovery. And then you'll notice I have one little one off to the side in orange. It came out of one of our control plots. It's very unlikely that, I think we're at 3 weeks post-release here, that a larva would have been able to crawl so far that they would have made it all the way into our control pots. It's the nice thing about releasing larvae is they can't fly. And it's clustering separately from everything else, indicating that this is very likely one of our native Chrysoperla species inside that Chrysoperla floribunda species group. So this is allowing us to separate all of our lacewings in terms of both being able to tell the treatments apart, but also being able to tell our released lacewings from what's already endemic in an orchard. So that's the method we're using going forward. So in 2022, we continued this work and we added in a drone-based treatment for the C. rufilibris, releasing eggs by drone. And we added, and then we removed that externa treatment. As I mentioned, we didn't think that that would be a very good climate match, so we stopped testing it. We added in the C. rufilibris egg cards, and as you can see, this orchard had a really nice population of woolly apple aphid. The release dates were slightly different for the 2 species. This is the other fun / challenging thing about doing release trials, is sometimes the US Postal Service gets involved with your research plans and you accidentally have delays and things get stuck in places. So we had to do a second order and have them come in a week later. So what we found in this trial is nothing. We found nothing. None of the treatments worked, which is incredibly disappointing because we thought this is going to be the year that we really be able to tell the differences between these different methods and whether or not one works better than the other. So why is that? I have one big speculation as to why that is. So you'll notice that we have 1.7 colonies per shoot as our starting number for aphid count. That is incredibly high compared to the year prior. And I don't think our releases were able to get going soon enough to really get the jump on controlling this aphid population before it got bad. So too late, maybe we put them out too late. There was also a lot of pest density variability between plots in this trial because we had to spread it between 2 different blocks of apples that were spaced fairly far apart. This orchard also used, although it was organic, more insecticides than the year prior, which used none other than some very specific codling moth products like virus. There was also more fire blight control product used in this orchard by quite a bit. Again, we've got those suspicions about fire blight products. However, we did recover our lacewings in this trial. And what was really nice is we actually found a few of them without trying. They weren't on our cat samples. We just walked through and someone observed one of our rufilibris, which you can actually morphologically identify with the naked eye if you've got decent eyesight and a nice hand lens, and then chomping around on a nice aphid colony here. And actually the recovery was quite good in this year. So what we found is the 2 best treatments for for recovery of larvae were the rufilibris egg cards and the rufilibris larvae. We did not ever find any lacewings in the drone treatment, with some concern there that maybe because lacewings as juveniles can't fly, that you have a bunch of eggs that don't make it into the canopy, they hit the ground, where they're either maybe eaten or the lacewings are just wandering around in the ground cover and they're not up in the tree. And then what was really cool about this trial is in the C. rufilibris larvae treatment, we found one larvae 8 weeks post-release, and there is absolutely no way that it was a larvae that entire time it was in the orchard for full 8 weeks, which means we're likely seeing a second generation of lacewings coming out of this release. So even if C. rufilibris can't establish, it looks like it can reproduce in orchards. And that's supported by the fact that we did find 2 C. rufilibris adults, although not in the plots where we put them in, because again, these adults are very, very dispersive and they started bopping around. We have not, as I mentioned, done the genetics work yet on our recovered lacewings, so we can only do the ones in terms of recovery that can be morphologically told apart, so the Rufilibris. This one could either be a resident or a released lacewing. If it is not a released lacewing, that means we recovered no C. pluribunda this particular trial. So this year, one of the other reasons that this may not have worked is our lacewings are actually put out later than the residents based on our release timing. So we were starting to find resident Chrysoprilla in the orchard the day that we did our pre-counts. So they were actually out first. They were also quite a bit more abundant than our released lacewings in terms of per plot counts, especially if we take Chrysopa into account. There were a lot of Chrysopa in this orchard. So in general, maybe we didn't get a head start on the aphid counts. They were really high. We didn't get a head start on the natural biocontrol present in this orchard. And also there were just more of them than what we put out. So in 2023, what we decided to do is use that same location because it had such a good woolly apple aphid population, but we moved it into just one section of the orchard and put our plots a little bit closer together because we hadn't really seen any risk of the larvae moving between plots. So that we could kind of keep it all in one variety, hopefully decrease the amount of variability we were seeing between plots and maybe make statistics a little bit easier and see some treatment differences. And then we did our releases on May 11th in this year. So here we were able to add the C. carnea or C. pluribunda available as egg cards at this point. And we removed that drone treatment and just focused strictly on ground since we had to keep our treatments so close together. And unfortunately, once again, we saw nothing. There were no differences between treatments in terms of aphid counts. You also notice our seasonal sums are a lot smaller than the previous two trials, but that's not because there were fewer aphids, it's because we only did the trial for 4 weeks instead of 8. The pre-counts were still kind of higher than I think we need them to be to see really good success. So we're at about 0.6 colonies per shoot. So we're getting close to having one on every single shoot sampled. I think that needs to be lower for this to work. And recovery was actually quite poor this year. So we only ever found 2 of the C. Rufilibris larvae. And that usually is the treatment where we have quite good recovery. We never found any C. Rufilibris adults. So we also only went out for 4 weeks. So it's possible if we'd gone out a little bit longer, maybe we would have seen them. But with that level of recovery, I'm a little skeptical. In terms of the C. pluribunda numbers, these could potentially just again be all resident lacewings. We're waiting on the genetics work to distinguish them apart. But based on what we're seeing down here for resident lacewing counts coming from the controls, I think this is just background noise, and odds are we actually recovered no released pluribunda either. So this is what I would call kind of a bad recovery year compared to the other two. And then also, again, no head start on the resident lacewings. They were kind of out and about at the exact same time that we got ours out. And there were more resident lacewings per plot than what we put out per plot. So have we learned anything, or am I just beating my head against the wall as I'm doing this with lacewings over and over and over again? I think this can work. And the reason that it works so well in that first year is because that site was much more northern. We were pretty close to the Canadian border there than compared to the trials we did in last year and this year. So I think that first week of May date might be great for northern sites, and we need to go in even earlier for our more southern sites, both because we need to get a head start on the natural enemies that are already in the orchard for this to work, but also because the aphid counts are so much lower and give those lacewings a shot at actually keeping them suppressed. This also might be an indicator that lacewings are a tool that really should be focused on more in orchards that have poor resident lacewing populations for whatever reason. That orchard that it worked in well had a really great spray program in terms of being very minimal and very selective. So I don't think their poor populations were due to sprays. It was something else was going on, which means those are kind of the organic orchards that might be good candidates for this to actually see some good efficacy. There's a little bit of an indication that releasing larvae might be our best choice. The price difference is not huge and our recovery levels are better. These results did kind of continue my drone skepticism, but I do want to have the caveat there that I don't think any of our trials were well set up to test drones just because the plot sizes were so small and we only sampled down the centers. So if there's even slight drift, we might just be barely missing sampling from where the drone hit. So to really test drones, we're hoping to next year and year after start doing really large scale whole orchard plots, where we're essentially treating multiple acres and then sampling straight from the middle to do a really fair test of that drone. I also have my eyes on organic bactericide, fungicide products and other organic products that are used early in the season. We need to do some work on whether or not these are acutely toxic to lacewing eggs and larvae, and then if so, how long those residues are harmful, so that growers have information about timing out their spray programs with when they're doing their releases to cause minimal harm to these insects that they've actually invested a lot of money in. And then the differences in the lacewing data from these site to site really indicates to me that we need to do multi-site trials within a given year, so that if you've got one that fails, you can kind of distinguish, okay, why is it different from one that in that exact same year didn't fail? And really get at the heart of the matter of getting these best practice recommendations really nailed down. I wanted to very briefly mention some other work that we've been doing on natural enemies and retention. And just kind of hint at it, and we can chat more about it if somebody's interested later. So what we've been looking at is whether or not we can increase both retention of released natural enemies, but also recruitment of resident natural enemies by doing what we call ringing the dinner bell, but also providing a snack. So it's known that many natural enemies are attracted to methyl salicylate lures, which are commercially available. These are plant defense compounds that kind of signal, hey, there's something that you should probably eat here. I'm being attacked. And the natural enemies can eavesdrop on that signal and use it to figure out where pests are. And there are food products that are commercially available that are commonly used in greenhouses for providing natural enemies with some additional food supplementation that we've been trialing, with the most promising one looking like brine shrimp cysts or Artemia tape. So we're looking at seeing how that's going and kind of doing some molecular work and on the final end of that 2-year trial right now. So stay tuned. So final big picture takeaways from both the work I've shown you and some of our additional release work is, first up, this is really hard. This is not like when a new product that's a pesticide hits the market and you test it in a few locations at some of the industry recommended rates, and pretty quickly, sometimes even within a year, you have an answer of how you should use it in your crop. It's much more complicated than that because you're dealing with living things. It also, and that's from the research perspective. From the grower perspective, it's hard to do this because there are just some years where it's not going to work, and sometimes you aren't going to be able to figure out why. So this is a hard tactic for growers to use, and I applaud the folks that are trying this as a way to really maintain their organic programs. One thing I really like about doing releases that can be challenging when you're doing it when you're talking about conservation biocontrol, frequently you're just telling a grower, don't spray, and the natural enemies will show up and your pest problem will be taken care of. And that is a really difficult thing for somebody that is rightfully risk averse to do. Say, just don't do anything and it'll all be fine. So this can be a tactic that's used that does not disrupt conservation biocontrol, that gets at that I need to do something without being harmful to your resident natural enemies. And then finally, my big takeaway from the last several years of doing all of these releases is that recovery of your released natural enemy is not a measure of success. You can have a trial where you barely get any of them back and they appear to have actually controlled your pests. And you can have a trial like our 2022 lacewing trial where you get quite a few of them, relatively speaking, back and you see no pest control whatsoever. We've got to be measuring both as growers that are testing to see whether or not this works and as researchers and really focusing in on pest densities. So our next steps is we need to do some large scale drone trials. We need to do some multi-site simultaneous trials of the same treatments within the same year. We need to be looking at organic pesticide compatibility. And we also need to start testing more frequent within a growing season, lower rate releases to get more at what a grower would be doing and whether or not more frequent small doses of natural enemies might be more effective at controlling pests than just one big go once. with that, I'd like to thank especially all of our funding agencies that I have listed on the right, and folks that have provided in-kind labor and in-kind donations of natural enemies, and especially our massive technician team. These are incredibly labor-intensive projects, especially the molecular work on the back end. And just a huge thanks to the two folks that do a lot of the groundwork on this, Erica and Danny, and our many grower collaborators that have allowed us to play around in their orchards and wander around collecting lacewings and mealybug destroyers.
Outstanding. We have a few minutes, not many, but a few minutes for questions. If anybody's got questions, go ahead and type them in the chat or take yourself off mute and fire away. I'm not seeing questions. All right, well, apparently you covered everything everybody needed to know, which is impressive. Oh, wait a minute.
Hi, Rebecca, it's Lynn Oaks.
Hi, Lynn, how you going? How's it going?
How are you? So my question, you mentioned Carnea may have been a better predator than the others you had available, but they're only supplied from the UK.
So Carnea, so it's not that Carnea is only supplied from the UK. It's that the real C. Carnea species is from the UK. So what's on the market is sold as Carnea, but is not actually Carnea, which makes it tricky to say, okay, this is a better climate match, because the research that was done on that, we don't know which species they actually tested. And it also depends upon what a particular orchard is selling as C. Carnea, because that can be several different species. So there's indications that if it's C. pluribunda, it'll probably be a better climate match. But if it's something else like the C. externa that we got by mistake, that's not the case. So yes, sometimes is like it's a very hard answer, but it's yes sometimes, and that's why.
Maybe one last question. Is anyone using biocontrol with beneficial successfully in apples in Washington?
Yes, and there are a few of them on the call. So Zirkle Fruit is one of the larger companies that's doing this. Gebers is doing this. And again, it's a question of what successfully means. The growers have a lot of other things to do and can't, measure to the level we're measuring. But their sense is that what they're doing is working, and they're showing that's how they feel by spending money on it every year.
Thanks for the update.
Thank you for the questions.
Excellent job.
Questions real quick in the chat. Parasitoids. Any other parasitoid species that might be effective?
So there is for mealybugs, there is anagris on the market. It's not specific to our mealybug. It's a little bit more of a generalist. So maybe parasitoids are really hard to release and assess well because they're so tiny. So it's a possibility, but it's not a strong one. And then on the aphid side of things, none of the aphid parasitoids that are on the market attack apple aphid, and we want something like, our best woolly apple aphid parasitoid is Aphelinus mallee, and it's just not commercially available. So people can't buy it in mass and release it. So there's just nothing for that right now.
All right. Were you tracking degree days at all with the release and capture dates?
I was not. That's kind of a list that I have on the back burner of to do. But for sure, just knowing what I know about temperatures in the two lacewing locations, the degree days would have been a lot lower in that first successful trial than they were in the other two. Okay.
And then finally, is Rhophilibus more arboreal than C. carnea?
So Rhophilibus is definitely more arboreal. With C. carnea, again, it's going to depend upon which one of the actual species in the species group you're talking about. If it's C. pluribunda, which it most often is, that one tends to be more of a scrub brush sort of lacewing, whereas some of the other members of the pluribunda species group, which are likely the ones we have in tree fruit, tend to prefer trees. So that's kind of another reason to really get at, if you're spending the money on C. carnea, who you actually have, because they have different habitat preferences too, amongst all the other complicating factors.
Yes, very many complicating factors. All right, well, we've gone over time. I'm going to let everybody go, and I appreciate it. Rebecca, thank you very much. This was one of the best attended and most questions, and excellent presentation. And I will just give a plug for our next one, which will be in February. We're skipping January. And on February 14th, we'll have Brittany Barker from the Oregon IPM Center on modeling emerald ash borer phenology under climate change. So again, thanks everyone for coming. Thanks, Rebecca. I appreciate it. And we'll see you all next year.
Thanks for having me, Steve.
You bet.